As mentioned in a previous lecture, proteins are so big that they can fold over and bond with themselves. Yes, yes, they can. Proteins are huge molecules with very complex structures. These structures are usually very specific and meant to be used for one purpose. It might be a stretch for two. I have some of the common uses of proteins here, but there are way more than just these 8 uses. You may have been aware that proteins do such things as serve as your muscles, or they serve as the structure of your hair and your tendons. We have had a mini lecture about how proteins serve as enzymes, but proteins are also the basis for your antibodies and also for some hormones. To us in this class, the most important uses of a protein are going to be as ion channels and as protein receptors. We'll explore these applications in detail. There are 20 total amino acids in this world to make all of the different proteins in this world. That's kind of a small alphabet and a lot of words. A protein is 2 or more amino acids bonded together. They are bonded by a covalent bond, but this bond gets a little bit more of a specific name. We call it a peptide bond. A polypeptide is a string of amino acids bonded by peptide bonds. This is not officially a protein, but it's the start of a protein. These amino acids are bonded together in dehydration reactions, in which water was removed to build or synthesize a larger molecule. This could also be called anabolism, the building of a larger molecule. I think of protein structure like those folded paper fortune tellers we made in second grade. What's your favorite color? What's your favorite number? Right? And then you open up the fortune teller and it tells you who you're going to marry. I married Mr. Sherman. Despite your potential spouse, the fortune teller, it's an important analogy since it's folded specifically to create the final product, right? If you don't fold it right, it's not going to work right. In fact, there are different levels of folding to create a final product like a protein. And proteins are kind of similar in that they're folded 3 times to create a final complex and a very specific structure. These 4 levels of folding are known as the primary, secondary, tertiary, and quaternary levels of folding. And each time a protein is folded, there are hydrogen bonds holding the folds in place. We referred to hydrogen bonds in a previous mini lecture, and we did say that they can serve as intramolecular bonds. Well, here it is. I hope that you're starting to, I hope that you're starting to realize that everything in my course is connected, whether that's by design or accident. It's kind of how it moves. So the first level of structure would be a sequence of amino acids or a polypeptide chain. The secondary level of protein structure creates 2 common shapes, one that is called an alpha helix, which is what you see right here. This is an alpha helix. And I'm sorry, I'm drawing with my finger because my computer's not exactly correct today. And this is what is called a beta sheet. It's like I'm signing my name when they give you the Apple Pay thing, and you're like, I can't sign my name. So an alpha helix is a region of a protein it's twisted into a tight spiral. The coils of the helix are held together by hydrogen bonds. And I just want to note that it's not a double helix. It's a single helix, an alpha helix. Other regions of proteins can fold up like a paper fan, and those are called beta pleated sheets. The alpha helices and the beta pleated sheets come together and comprise the secondary structure of a protein structure that results from one event of folding. A given protein might have, I don't know, 3 alpha helices and 4 beta pleated sheets, while another has 8 beta pleated sheets and no alpha helices. The combinations are almost limitless, but most proteins have at least some of each of these structures. Once all the helices and sheets are coiled, folded, and locked into place by those hydrogen bonds, the polypeptide takes on a distinct 3-dimensional shape, and it's very different than that string of amino acids. This is what is called the third or the tertiary level of structure and the overall 3-dimensional shape of a protein. The fourth level of protein structure describes interactions among different polypeptides, right? So not all proteins have a quaternary structure. The big ones usually do. But for many proteins, their quaternary structure is their ability to bind or meet up with another protein. Some complex proteins are composed of more than one polypeptide chain, and hemoglobin would be an example of that. You know, I'm always disappointed at like the naming of biological structures. I just feel like there's this great opportunity to name them something wonderful and beautiful, but then we end up with words like globular and fibrous. I mean, at least they're descriptional. But these two terms, they do mean exactly what you think. Globular proteins are a glob. I guess you could roughly say that they're spherical. Most globular proteins are water soluble, which means that they can be soluble in your blood. And most globular proteins have some very specific function or action, like helping to make ATP or being an enzyme. Opposite to globular proteins are fibrous proteins, which are strand-like. You might be familiar with collagen or keratin from purchasing beauty products. These are mostly structural proteins. They don't do anything more than just exist. Instead of catalyzing a reaction or being a channel for ions to flow through, these fibrous proteins, they contribute more to the structural building of the human body. So we see how the protein structure is made through these levels of folding, but we can also deconstruct a protein. This process of changing the protein shape or unraveling it, unfolding it, is called denaturing. And denaturing usually involves a chemical, like an acid, or heat. I love me some eggs. I do. Scrambled. That's how scrambled. When I cook the eggs, I'm denaturing the storage proteins in the egg. When you are denaturing meat, you can do that with a marinade, especially an acidic one. You can denature meat on the barbecue with heat. We can actually denature proteins, making them safe for our consumption without using heat. Fish exposed to acids are fully cooked, and this is what is called sous vide, making the fish safe to eat. So eggs that are exposed to lemon juice are actually safe to eat as well, because the lemon juice cooks the egg proteins much like the heat from my frying pan.